welcome back. We're going to start our class, and we had our quiz on Tuesday, our first quiz on Tuesday, and I graded that, and it was worth 20 points, and so on your quiz you have a circled score, how many points you got out of 20, and I'll hand those back at the end of class. And let's see if I have a slide about this. Yeah, and we'll just go to this for now, and that is, we have a new thing here, and that is, so when I grade quizzes and exams, the way I do it is I typically just mark what points are taken off on the quiz, so you'll see that today, and I rarely write on the quiz, because what I do is after grading the quiz, I make a video of myself talking through the quiz like I'm grading it, and in doing so, I can help, because I just looked at all the answers the students gave, I can help everybody by saying some of the wrong things, of course anonymously, but not some of the wrong answers and why they were wrong, right? So think about that. Instead of just sharing those explanations with the individual student, a lot of times it's super helpful for everybody. Even if the student got all the points, it's helpful to underscore, and it's like, coaching like that, and trying to coach the whole team, right? And so I will post today this answer key video, and it's really good for everybody to watch it. I try to make it short and to the point, but I try to make it so that it would help everybody to see that. A lot of times it's helpful to see a student that lost a point because they didn't have it quite right, like that. And even if you've got all the points, you're like, okay, all right, I see that, and it kind of adds to your learning. And that's really a big part of any kind of quizzes and stuff like that, is to try to loop through that learning and strengthen it. So I'll post that, and I do that each time. And after that, you might still have questions, issues, and stuff like that. And you can bring them to class, that's great. And it could be like, oh, I added them up wrong, or I marked them wrong, or you want to clarify something that you thought was a pretty good answer, or you're just like confused about the explanation still. Absolutely. So we're going to follow up on that. Because this first quiz is considered, we would hope that you would bring that to this course, right? Alleles and orthologs, but you don't, because students took intro biology from all kinds of different people. So we shift around, right, and have that as as we move forward, what we want to get done this week. So that'll be up on Brightspace along with the recording today. And so again, we're just gradually going to be grinding through chapter 1. And I completely failed to put this on this week, is that because today we're going to start talking about chapter 6. So I showed that last week, that our first reading assignment was chapter 1, and the next is to jump into a few pages or several pages of chapter 6. And we're going to get to that today, and I just messed up. You should be doing that this week, is looking and reading chapter 6. It'll take us a little while to get through that, but and you don't have to have read it yet, so don't freak out. But I just completely spaced out about doing that. And and I'll remind you in the video. But I did have the reading up there last week, and to say that was going to be our next reading. And if you don't have our book quite yet, that material is still on that electronic resource, that chapter 6 material. So we're going to roll ahead with that, and then really importantly, we're going to have a quiz on these muscle differentiation videos that we've already talked about a lot, and those are really super significant and foundational in our course. We'll talk about them again and again and again. So undoubtedly, you'll watch those videos multiple times over the course of the semester. They'll be there the whole time. So that's where we're at with that. So let me just show you this to show you about, kind of help you about this idea of this answer key video, because I'm like a really friendly person and supportive, but when it comes to answering questions on quizzes and exams, they have to be pretty specific, and I'm not really fussy. You'll have some teachers that are all about, well, you spelled that wrong, or that's the wrong punctuation. It's not about that, it's about content. But it's you being able to express to me that, yeah, you understand what that's about, right? So here we have orthologs, right? One of the questions on the quiz was, what is an ortholog, like that? So orthologs are two genes in different species that are understood to share a common ancestral gene. So that's what an ortholog is, right? And we'll be using that concept throughout our course. So this was actually from last year. I gave the same question on a quiz. And so let's just look at some different students' answers. And again, these are always anonymous, so don't worry about it. Ortholog, genes in different organisms that share similar functions. So that's true, right? They are two genes in different organisms, and they have similar functions like hemoglobin and humans and dogs. But what that's missing is this really important concept that they're understood because of their similarity to share a common ancestral origin. That's a real key concept for the term ortholog. It's natural, oh sure, hemoglobins and humans and dogs are similar, but there's more to it than that, more to that to just have a similar function. Yeah, that's always got to be part of it. But they also have that similar sequence that's so astonishingly similar, we talked about last week, that you just have to say the only way you could be so similar is there must have been a common ancestral gene. Then orthologs. Orthologs are the same gene found in different species. So same gene is not, it is the same gene, yeah, that's true, and they're found in different species. But we also need that thing of their relationship, because that's what the big, that's a big part of this, is there's a relationship that's tying all life on our planet together in evolutionary relationships. Ortholog. So, and the thing was worth 2 points, so they're missing, they're missing a piece of it like that. So, and if something's worth 2 points, I try really hard not to say, oh, and you missed half a point. I'm like, well, 2 points or 1 point, like that, right? Because it's just become such a thing, where you're like that. So, ortholog. An ortholog is when two different species with two different genes share a common ancestor. So, that's another thing that is important to point out. And I, sometimes when I grade this, I'm like, oh my gosh, I, in my explanation, didn't emphasize this enough. And that is, up until now, you've always thought about evolution as being that similar organisms shared a common ancestral organism. Like humans and dogs, we're both mammals, and there must have been a common ancestral mammal, an early mammal that we're both related to. Here, we're not talking about the organism, we're talking about the gene. So it's really important to appreciate that we're looking at the gene, and we're no longer paying attention to the organism. You drop down to that gene level, and it's not that the two species share a common ancestor, but that gene does. And we'll talk a lot about that in our course, because we can look at organisms and compare organisms, and that's called phenetic characteristics, just similar characteristics. Yeah, we all have hair, dogs have hair. We look similar, we're probably related. Let's make an evolutionary tree based on those observable similarities. We dropped down to that genetic level, that DNA sequence level, and we saw that astonishing similarity based on that probability, right? That the dog and human genes are so similar that they could occur by chance only 1 in 10 to the 60th power. It just brings in a striking level of evidence. You can think about it like this: when you compare a dog with a human and you say, what are similar? Each time you say something that's similar, that's a piece of data, piece of evidence. So hair, they have womb, like that, boom, boom, boom. And you can just get lots and lots of similarities. And think about how many you can get, let's say you can get 150, like that. Well, when you do the sequence of just one gene, you get astonishing amounts of data, because you can do that for that gene, hemoglobin, and so forth and so on. So that student lost a point because of that they weren't saying it was really the ancestral gene. And I know that sounds kind of fussy, and I don't think I emphasized that well enough, before like that. But we want to emphasize that. And of course, it's just 1 point in a quiz, and that's why we have quizzes. It's to be like checking in for not just the student, but also the instructor. Then the last one, orthologs are two genes in a different species that share a common ancestor. So great, they got 2 points like that, okay? And so I don't use photos of examples on the when I'm making the answer key video. I'm just talking about it, writing down the answer. But I talk about those different ideas, trying to help everybody out, So, really, you really want to watch it. Often I make an assignment where you have to watch it and do stuff, but we're not doing that. It's a quiz like that, okay? So we're going to talk again about these two scientists that took advantage of the idea of genomic equivalence to turn any cell into an induced pluripotent stem cell. So it's a lab technique like that. And talking about it today, let's just do this to start out with. So, I didn't pull up my pen yet. So we're going to talk about stem cells. And our book talks about stem cells in chapter 9. But we want to start our course with it because in our muscle videos, we have a stem cell. So there is a muscle, and it's every kind of long, striated muscle fiber in our body, and there's the muscle, and with its contractile apparatus, so that's the muscle differentiated cells. And around the lining, we have these scattered cells that are reserved. They're called muscle satellite cells, and lots of people, I always might forget how many T's and L's there are in these words. Satellite cells, they're a type of stem cell. And that's in the news a lot. These are a type of stem cell that's called an adult stem cell. And that's because we have them in our body right now. Adult stem cells are a misnomer because you have them in your body when you were a kid, and when you were a baby, and when you were a fetus. So they're in our bodies right when your heart starts to beat in your mom's tummy. There are already some of these adult satellite stem cells. And people just lump them together because they're maintained. We have them a different set in our bone marrow that are constantly making new blood cells. We have them in our skin, constantly replenishing our skin through our life. And how do they do it? And the answer is when these stem cells divide, they divide in a distinctive way, where when they divide into two cells, and one of the cells continues to be a stem cell. And then the other cell is going to not be a stem cell. So it looks like that. And so these stem cells are said to be self-renewing. And that's a great term because you see how they're self-renewing? We never run out of them. They get activated in our bodies. You get activated today if you get injured and the muscle has to heal, need some more muscle cells. Or if you work out and your muscles grow, much of that growth is due to activating the muscle satellite stem cells. Like that, or when we were kids and just naturally growing up as getting bigger as kids, bigger muscles just from growing a bigger body. So they get activated and they divide, but we don't lose them because they divide in that self-renewing way. Okay, so that's how all adult stem cells divide. Is they're self-renewing and they divide unevenly. And the other cell is just because it could be in all kinds of different tissues. We just call it a precursor cell. And in the case of the muscle satellite stem cells, all that can do is can grow and turn into a muscle. And so what we know is that the muscle satellite stem cell can divide, make a precursor, and in this case, that precursor becomes a myoblast, a muscle precursor, and eventually differentiates into muscle. Yay. So that's how that works all the time. It's just what is the tissue and what are you going to be making, like that, okay? So now check this out, right? So we have this cell division, right? And so see when the satellite stem cell divides, it makes two different cells. Anybody happen to remember what kind of development that is? Because there's a term for it, because this is one of only two ways that cells can become different in life on Earth. And it's called,
Is it just differentiating?
Yeah, that's a good question, or comment, and no, it's not differentiation. Differentiation is when the cell reaches what it's finally going to become, yeah?
Unequal cell division.
So this is a type of unequal cell division. And that's really important for us to notice because that's one way for cells to become different. There are just two ways. Either the cells divide unequally or they communicate with each other to become differently. And there is a specialty old school term that I'm pretty sure I mentioned, but just once, and the book mentions it too. So this term is a good example of where something came up in class, but just boom, like that, just one time, like that, you know? We'll say it over and over again, but just one time so far. The book uses it too, so in looking at the book, you would also pick this up, that it's a common term. So, I hope I mentioned this, I think I did, but just like one time, it's called mosaic development. And so that's one way that cells can become different, and that happens, and there are many, many examples of mosaic development in all animals and plants. Okay? Now, in the context of stem cell biology talk, and some other types of biology, this unequal cell division or this mosaic development, the stem cell biologists don't use that term. They say, and it's a good term, asymmetric division, unequal or asymmetric. So that's just a specialty term. It's synonymous with unequal, and it's just in the context of stem cell biology. They say the stem cell divides asymmetrically. It's self-renewing. It's dividing asymmetrically to make itself again, self-renewing, and a precursor cell. Okay? So that's the setup. All right? And maybe just before we go back to that, I will just say that when we talk about stem cell biology, we could talk about adult stem cells, and there's another kind of stem cell, embryonic stem cells, that are only found in mammals and only in very early embryos. And then today, we're mentioning that in laboratory, these scientists around the world can now make these induced pluripotent stem cells. So those are the types, the categories of stem cells that we could talk about in animals. Adult stem cells are in us, embryonic stem cells, they're only in really early embryos. In humans, just the first week of a human pregnancy are embryonic stem cells there. So those are naturally occurring stem cells. And then we have this one that's made in a research lab. So induced pluripotent stem cells, or they're abbreviated IPCs, induced pluripotent stem cells, like that. Okay, and so that last one is the one that was like invented, created, produced in a lab by adjusting the expression in genes. And so we're going to talk about that, okay? All right, so now we go to this. Remember, this is a very specific example, but it also gives us a general appreciation of genes and gene expression, okay? So remember that we are talking on stem cells. A cell just divided, and the two daughter cells are going to be different cells. Stem cell and a precursor cell. And that Yamanaka said, oh, I should compare those two cells to see how the stem cell is different than the precursor cell. Because the stem cell is going to continue to be a stem cell, and the precursor cell is going to stop being a stem cell. So what does it mean to be a stem cell? What does it mean to stop being a stem cell? In development, that almost always involves changing gene expression. Yes, the stem cell has the whole genome. Yes, the precursor cell has the whole genome. But they start to express different genes. And we can study that by studying the RNA. And so Yamanaka just studied the RNA of all the RNA, all the genes being expressed in a stem cell, and compared it to that that's being expressed in a precursor cell. There must be some genes that are expressed in stem cells that are somehow making it be a stem cell, and they get turned off in the precursor cell. So you can do the experiment and detect the RNA for those genes in the stem cell and not detect them in the precursor. So that's the idea. And it's based on this, two cells, myoblast and muscle. This is not a muscle, this is two cells that are different. In this case, just a little different in time, you know, one is just a day older. So how are they different? And we know that changing gene expression, and we know how the gene expression is changing, right? So again, genomic equivalence, the myoblast and the muscle have all the genes, all the same DNA. Somehow the muscle is different, and it's really different than the myoblast. It's got all those muscle genes expressing, and so we can study the RNA in these two cells and make the comparison. And we can say, oh, I see all these muscle genes being expressed. And then in the case of the video, we say, how did those get turned on? You know, and the answer is through the regulatory regions of those genes and transcription factors binding to them. And then we say, as developmental biologists, we're really interested in how the genes get switched on. And the answer is, well, it's through these transcription factors. So developmental biologists almost always are going to, well, what are those transcription factors? And the answer in muscles is MyoD activates myogenin, and myogenin activates lots and lots of these muscle genes. So we get a big shift in the types of genes expressed and the types of RNA in the cells, and that's huge, like that. So it's a huge example, and that's what Yamanaka is thinking about. He's thinking about, like, I see that happening in other cells. It must be true in the stem cell story. So let me study that. Yes?
I was gonna ask, so here you said that the precursor becomes a myoblast, and so which one of those two cells would be the remaining stem cell?
Yeah. So let's just go back to this and just show that a little bit. So I'll erase this, and because I drew it down there, and you see it gets pretty messy. So here we have, and we'll just draw this out. So here we have a muscle satellite stem cell, linear muscles, and it's just resting. Right now, you know, they're just stopped. They're stopped in the cell cycle. And in intro bio, you should have learned something about the cell cycle and know there's G1S, G2M, and this is just sitting at start. And something's going to stimulate it to go through the cell cycle. And there's just such freaking exciting biology, because you know what activates these things to start to grow and divide? Mechanical stimulation. So think about it. When you're a kid or you're working out or you're injured, always what's going on is, a muscle has to work harder. You know, if it's injured, it's like I missed part of it, and this remaining cells have to work harder. And so somehow that mechanical stimulation is actually in part sensed, right, and converted into some changes in biochemistry, right? And it's really not this cell that's doing it. It's other cells, like in the bone and in the rest of the body area and stuff like that, that are communicating with it and saying, yeah, you know, we need more muscle. So again, I said before, you know, squirrels, we should just be out there petting them without fear. And being like, hey, little buddy, you know, you're doing great. Because they have muscles, or any insect, you know. So this gets activated to divide, to go through the cell cycle. So it goes through the cell cycle. It might go through it repeatedly. So it's going to divide like this, and it divides. There's a stem cell. There's a stem cell. So that's always happening. It divides asymmetrically, unequal cell division, mosaic development, and it's self-renewing. It's going to maintain that stem cell status. So then this is a precursor cell. And that's always the case in whatever the adult stem cell is, whether it's in our bone marrow or brain, skin or whatever. So then these precursor cells can, in different ways, divide, because sometimes they increase their population quite a bit. So they could go through some serious development, but they're going to become a myoblast. That's what this can make. It can make muscles. And on the way to be a muscle, you're a muscle precursor. So that's the answer to your question. Is we got a stem cell, then this precursor that's, you know, it knows where it's going to go. And through a specific developmental pathway, it becomes a myoblast. And then it differentiates into a muscle cell. So these will all differentiate into muscle cells. And along the way, there's a series of genes that change in their expression. That's why we said, in a myoblast, first we activate MyoD, then we activate myogenin, then we activate muscle genes. It's always this cascade of gene expression, one gene after another, as the cell moves down that pathway. So a stem cell is different than a precursor cell, it's different than a myoblast, it's different than a muscle. They're just changing gene expression all the way. lots of times students will say, well, what activated that MyoD gene? And the answer is, yeah, there's a story upstream of that. What we're choosing is, we're imagining like a series of dominoes falling, and each domino is a gene, so that's an analogy we use a lot. You know, people will set up dominoes and they knock down each other like that, big cascade of them, right? And all we're doing is we're saying, to learn about that, generally, we're just gonna grab 3 dominoes and watch them fall. And yes, there was a domino that knocked MyoD down, but we don't need to talk about that. 3 is enough to get on board with this idea that we're gonna activate MyoD. That means we're gonna start expressing the MyoD gene. It's always been there since fertilization. There was a MyoD gene, one from mom, one from dad. Now it gets activated. What does that mean? There's transcription factors binding to the regulatory region, and it's starting to get transcribed. So we can now detect MyoD RNA. It's a coding gene, so it makes mRNA, and we can start detecting MyoD protein. And MyoD protein immediately goes into the nucleus to do its job, its function as a transcription factor. It binds to the next gene, myogenin, which is the next domino. And that's what we mean by the MyoD knocks over the next domino. MyoD activates the myogenin. Then myogenin starts to get expressed. The DNA was always there, now it's going to express. We can detect myogenin RNA. We can detect the MyoD RNA a little bit earlier in time than we can detect the myogenin RNA. But pretty quickly, we start detecting myogenin RNA. And that's a coding gene. It codes for myogenin protein, so that's an mRNA, messenger RNA, and it gets translated. Now we can detect myogenin protein. It immediately goes in the nucleus to do its job, which is to bind to the regulatory region of muscle genes, like muscle myosin, muscle actin, and so forth. Activate those, now we can start detecting those. Those are all coding genes, code for real muscle proteins, like that. So it's a cascade, and we just grab that. So what you asked is that's what's happening. And the significance of that is we drop down to the gene level, we say, oh, as these cells are changing and progressing, they are changing their gene expression, like that. And so we'll grab little portions of those stories to tell the principles. And that's what we've done in these videos. If we just grabbed a little segment of the developmental story to talk about the 3 genes in that cascade. And something is like that is happening in the stem cell when it divides. The stem cell maintains expression of some genes. The precursor turns off that expression. So it's just the flip of what we were talking about. We were talking about a cell becoming a muscle cell. Now we want to talk about a cell stopping being a stem cell. And so Yamanaka is just like, okay, well, let's look at the RNAs in the stem cell and look for a gene that is not expressed in the precursor. And so they looked for those, and they found, initially they found 21 genes that were differentially expressed. And so then they said, let's do a genetic manipulation where we force the precursor cell to maintain expression of those genes, and find out which genes are required for being a stem cell. And people call that stem cell identity. And which genes are required to be expressed for a stem cell identity. And so the way they did that experiment is they just took these cells and they manipulated those 21 different genes by changing the regulatory region. So the regulatory region wouldn't turn off the gene, it would maintain its expression. And when they did that, they discovered that they only needed to maintain expression of 4 genes to maintain stem cell status. And just to draw that experiment out, what it looks like is this, that in that experiment, those cells are dividing, and the result was that these cells didn't become precursors, they became stem cells. And so they would divide, and they would make 2 stem cells. We were just maintaining the stem cell. We're not letting the cell stop being a stem cell, like that. So it's experiment in lab, you know, and they're just like, woo-wee. All the experiments were done in just 6 years, because they were just like, they had the tools and they had the idea. And you can see the idea is not like, oh my gosh, you know. They just went for a walk and they said, well, if that story is true about muscles and all other things, there's an implication for what is happening in the stem cell and the precursor, right? So they just found these 4 genes, and if they maintained that status of those 4 genes, they would maintain this stem cell, and people call it stem cell identity. There are genes that need to be expressed for a cell to be a stem cell. And we'll talk about it more when we get to chapter 9, but just for today, let's just say a couple more things about it, right? So here's one. All 4 of the genes are coding genes that code for proteins, and all 4 of the proteins are these transcription factors. And transcription factors are proteins, and their function is to activate other genes. So these 4 stem cell identity genes can activate a bunch of genes. It doesn't take 4 genes to be a stem cell, it takes a bunch. But these 4 can activate all of the stem cell genes. Like that. Remember I said at first there were 21? Well, these 4 activated those and more. Like that. So that's a key concept that we're picking up in this muscle differentiation video, is that these transcription factors are very interesting because development is all about changing gene expression, right? We always have genomic equivalence, we have that DNA, so it's changing. So that's one thing, is that they're transcription factors. And then the next thing is those transcription factors are said to have target genes. What does that mean? And we use that term in the muscle differentiation video. MyoD binds to myogenin, and so myogenin is a target gene. In our example, we just show myogenin. MyoD can bind to lots of genes. There's lots of MyoD molecules in the cell when it's being made, and they can bind to all the target genes. And so that's typical that a transcription factor has several target genes, and it just activates them all, or represses them all, depending on its function. Okay? All right, so these transcription factors do that. And so then we would say, well, that's interesting. We see that they're transcription factors. They're going to activate those genes to have them be stem cell identity genes. What do the target genes do? What's their function? And their function is to maintain the stem cell status, like that, okay? And the function comes up really dramatically when we hear the last piece of Yamanaka's discovery. The last piece is Yamanaka's lab found that they could force the expression of the 4 genes and maintain stem cell identity in his precursor. That's great. It's not too surprising because it just was a stem cell, so that's too surprising. The big deal is that the Yamanaka lab found they could force the expression of those in any cell, and it would become a stem cell. And that's just startling, because remember, the muscle has gone through differentiation. It's gone through a lot of changing gene expression, and it's a muscle, and it's not changing. Lots of people call differentiation, they call it terminal differentiation, because it's the end of the road. That doesn't mean it's going to die, right? Sometimes a person has terminal cancer and they're going to pass away. It means the end of the road. Our brain cells have all gone through terminal differentiation. We're going to have them the rest of our life. They're not dying, they're just done. And that's really typical in animals, that when a cell goes through differentiation, it's going to keep that for the rest of its life. It's not going to change. That's really, really typical. That's a famous common feature. A lot of times people think that's done to avoid cancer, because when a cell goes through terminal differentiation, it's going to be that, and it's not going to divide anymore. You know, in our brain, we're not getting more brain cells. They've terminally differentiated, and they're done. They're going to survive and live for the rest of our lives. We're not going to get more. They're not going to go through the cell cycle more, like that. That's why we need muscle satellite cells, because the muscle tissue cells have differentiated, and they can grow, they can get bigger a little bit, but it's limited. If we need more muscle cells because of injury, childhood growth, or working out, we need a source of cells. So that's why we have those stem cells lining our cells like that, right? So Yamanaka's discovery that they could misexpress the stem cells identity genes in a muscle and turn it into an induced pluripotent stem cell is just remarkable, because that almost never happens in nature. There's another thing, remember, in the videos, if you've watched them, hopefully you've started watching them, that we say when a cell differentiates, it still has all the genes. Like those muscle cells, they have the insulin gene, but they do this process of DNA methylation, and they methylate the genes they're never going to use again. They're done, they're not going to be another kind of cell. They've stopped the cell cycle, and they've methylated a lot of genes that they don't need. They hang on to them, they don't need them. So, last thing to say about the Yamanaka story for now is, when they looked at the target genes for the 4 stem cell identity genes, they looked and they said, what do those target genes do? So, not surprising, just super satisfyingly, they make the cell go through the cell cycle again, and they demethylate DNA. So they open up the possibility of expressing the insulin gene, the hemoglobin gene. It's there, but it's not going to be expressed because that DNA methylation always is going to repress genes. So we got to remove those DNA methylation marks, those methylated DNAs, and also get the cell back in the cell cycle. And there's some other things to do, but those are the big deal, like that. And it's just startling, but for us, the reason it popped up is because I guess I don't have the slide up here anymore. But the reason it popped up is because it's just a remarkable example of that genomic equivalence. That's when it popped up when last week we first started saying, oh, some plants can grow little plants off their leaves or cuttings or twins, And we said, it turns out that that's really common on Earth. And this is one of the most remarkable features of it. And it's rooted in that, right? This Yamanaka is like, you're talking about muscles changing into muscles, but still having genomic equivalence. We got the whole genome there. So in theory, Yamanaka thought, I should be able to change any cell into a stem cell and make the whole organism. And that's exciting because there's the possibility, I said before, of having regenerative medicine grow you a new kidney that's yours. Or just a new patch of skin from a bad burn that's yours. Like that. We had a student several years ago now who got to do a summer internship at the Harvard Stem Cell Institute. And he told me, he said, that's the only kind of stem cell they're studying because it's just so exciting. Down at Maine Health Institute for Research, there are a couple of labs there that they're really focused on those because they're trying to do this regenerative medicine. Different types of regenerative medicine. Again, different examples. Somebody got injured, spinal cord injury. Can we grow some stem cells? Let's have them be their cells. That avoids, graft rejection type thing, transplant rejection. All right, so wicked cool, and you can see, remember that example I gave that biology's awesome, but there's that giant sloth coming into our room, and that just derails. It just derails us. And that just derailed us. You say, oh, genomic equivalence, and I'm like, plants and stuff and twins, and then you go, right? And that happens a lot in biology because the biological nature of the Earth is that there's tremendous diversity and tremendous variety of biological stories, and a lot of them, when we hear about them, we're like, oh my gosh, we could take advantage of that, or that has an implication, and we should do some experiments. Because we could check it out and maybe create a product or a therapeutic strategy or just understand our bodies and life on Earth better. Crazy. So there's the thing, and we'll loop back to it. But it's a thing where we just kind of want to go there because it's so satisfying. As another example that complements the muscle differentiation videos and helps you go, oh wow, yeah, I can see why we're learning those. And that's an example of that. All right, great. So I should pause there and just check in about people having questions about this, that, or anything at all. Syllabus, quiz next week, like that. We'll have the quiz at the end of the class on Tuesday. So there'll be time to ask questions then, or, you know, anything from the book or anything like that. Yep. And you can blurt out questions anytime like that. I used to say, does anybody have any questions or comments? Because I just, for some reason, I thought that was cool. Anybody have a question or a comment? And there was a student named Heidi, no, I can't remember her name, Haley, several years ago. And Haley would stand up when she wanted to make a comment, like that. Stand up and kind of hold her chair and say, I have a comment. And I'll be like, excellent. Just great. I love being a professor because you are all so fascinating. You know, you're definitely all unique. Like, I'm just like, oh, get him, Haley. Yeah, it was just wonderful. So does anybody have a question or a comment? I'll make a comment. It is freaking nice outside. Oh my God. Yes, okay. So, and you can interrupt anytime, either by raising your hand or standing up or just saying, hey, I'm going to issue. And you want to just blurt them out because you know when you're thinking about something that's distracting you and you want to kind of clear your brain. All right, so this is chapter 6 stuff, and we're going to talk about 2 stories in class, and the book covers those in that chapter 6. And the reason we're talking about them is to sort of dive into our course and see what is it going to be like. And we're going to use this as an example. Our book talks about more different kinds of experiments and biology in this little nematode worm than we'll talk about. So I always bring my book because when you read chapter 6, you'll say, well, what do we have to know from this figure? Let me just find a page like this. Yeah, I marked it because here's a figure from the book, and it's a lot of the figures in our book are pretty simple, but some are complicated. And if you said, what do you have to learn about chapter 6, figure 617, I'd be like, nothing. So see, that's just a really great question, right? Because you can just say, like, I'm not even going to read it then. You're asked to read it because we want to spread out your learning and understanding, but the book is not equally important. One of the ways you know what's important is the examples we talked about in class, like that. And but there isn't going to be, like, you can ask some question, they'll be like, yeah, there's no way there's going to be a question on that figure on the exam. In other cases, I would say, yeah, you need to read it, you need to understand it. We could have a short, 2-point question about that on the exam, like that. Okay. So we're going to talk about these 2 stories. One of the reasons to talk about them is because they were awarded the Nobel Prize 20 years ago, like that. Okay. So the first one we're going to talk about is programmed cell death, and this is the species where that was worked out. And programmed cell death is when a cell dies during development, and it dies because it's like its fate. There's a plan for it to die. Examples of that would be our skin. Our skin cells die. Our blood cells die. And they don't die because they're old or injured or malnourished or lacking oxygen. It's part of the plan, like that, okay? And scientists knew that this was happening like 50 years ago, but it was really hard to provide like experimental evidence for what was going on, and it was really hard to really kind of believe like that, that a cell would be in good health and plenty of nourishment, and it would just die like that, okay? So here we go with that, and here's the reading assignment here, like that we're supposed to be doing, and I just failed miserably to have that on our week 2 work. So you're going to read that, and we're going to be studying one of a handful of organisms. So there's a good chance that you've never heard of this nematode. Nematodes are a type of worm, a real simple worm, that there are a few hundred species on Earth, and some of them have, some species cause medical problems, like a parasitic type disease problem, in their study. Some of them play pretty big roles in the ecology of certain environments, especially famous in just the natural soil environment. So there's some bacteria in there and stuff like that, fungus and stuff. And there are a lot of different species of nematodes. This particular nematode species is called Cyanorhabditis elegans, or people just call it C for its genus elegans. So C. elegans, or we'll just call it nematode. And it was just chosen as one example of a species to study, and that was done in the 1960s, where scientists just said, we should pick that. There's a lot of features of it, grows fast and so forth like that. Let's study that. And so it became what's called a model organism. And you know the idea of a model organism because you know that lots of lab research is done on mice and rats and fruit flies. And those are examples of model organisms. And in developmental biology, there are just a handful of model organisms that are they're studied in research labs, and they get studied a lot. And they've taught us, lots and lots of the foundational stuff we need to know. So here are some famous ones, and specifically in development. So the C. elegans nematode, then mouse, and then this flower. So we'll get to that in chapter, I think it's 13. We'll jump to that after our first exam. And people just call it Rhabdopsis. It's just a simple little flowering weed. There's a fruit fly. Then here's one I want to mention. It's a zebrafish. In any pet store that has tropical fish for sale, they have zebrafish. They're really inexpensive because they're really boring. They're just little striped minnows like that. But it's a really significant model organism. And throughout our book, the author is walking through the parking lot, turning to the different vehicles to teach you specific things about car mechanics. And it's not always cars, it could be a jet airplane, or a handheld electric mixer, or a mower, like that. And so that's what's going on here, and our author does that, right? And throughout the book, they use zebrafish as an example. And the reason that it's a good example in a model organism is because it's a vertebrate. So that means it's pretty closely related to us. Not as closely as a mouse, but it's a vertebrate that has external fertilization and development. So both the male and the female release sperm and egg. Fertilization happens out in the environment, and development happens out in the environment. And so that allows you to look really right away at embryogenesis. The mouse, human, other mammals have internal embryogenesis, really hard to study, like that. The other reason why zebrafish is that these non-mammalian vertebrates, like fish, birds, reptiles, they typically have really big eggs because the embryo is going to need nourishment before it grows and can feed itself like that. And you know what that means. They have yolk, like a chicken egg yolk, or like the yolk in the little amphibian eggs we see in the spring in the ditches and streams. The zebrafish just happens to have an egg and yolk that is perfectly clear. And so that's a great advantage because you can just watch it under a microscope while it's alive. You don't have to kill it and dissect it. It's out of the body and the whole show is there, like that. And so here's and then we saw there was this story, and we'll talk about this in some detail in our course. There's a story in the videos where I said that scientists can genetically modify a mouse and create a mouse strain that has muscles that glow. And so scientists can genetically modify a zebrafish and have muscles that glow in it. And you can just make a video of that zebrafish strain and just watch, and there's no glow. Fertilization, cells dividing early embryos, no glow, and then suddenly cells start to glow. And you say, there's the first muscle differentiation. So the zebrafish is just a powerhouse for vertebrate development because of the combination of clear embryo and the ability to tag cells and then watch them when they glow, like that. Oops, let's see where we are. And so that's great. So it's really fabulous. And lots of the professors across the country that teach this course, they study zebrafish. Here's the deal for us. We're just going to go over that very lightly. You're assigned to read it, but there's never going to be questions on our exams about zebrafish. We'll focus on other model organisms. We'll have great examples from them. And the book has a lot of zebrafish biology in it, but we just have more satisfying examples. Yep?
By glow, do you mean to glow under like some sort of chemical reaction or like actually just glow?
Thank you. No, just glow by illuminating it with a UV light. And the cute trick is humans can't see UV light, So it's shining on the zebrafish embryo, but we don't see it. And it's just exciting those tags, and they glow. And so to us, it just looks like everything's dark, and we get that glow happening. Yep, yeah, okay, yes.
So is that a real picture of a bacteriophage?
Is that a real picture of a bacteriophage? Yes, it's a transmission electron micrograph. Yep.
That's really cool.
Yep, yeah, and those are some simpler model organisms, right? That you may have heard of, yeast, bacteria, and stuff like that. Not really for development, right? Because we study multicellular plants and animals. And so some typical ones, mouse, fruit fly, and some ones that are really kind of ones that developmental biologists really pick on, like that. And so we're going to talk about this nematode, and it's a model organism. So this is a big part of developmental biology, is that it's being driven forward by studying a small number of model organisms in laboratory settings. So much has been learned that we can take that to other organisms, right? Because we learned a set of orthologs in fruit flies, and that allows us to study like any animal. So it's a real powerful strategy because of the relationship in evolution. So we go to orthologs. Orthologs are the basis, the justification for studying a model organism. We're really closely related to nematodes. You can see what's going to be happening. The scientists are going to work out the genes involved in the dominoes, the steps, I shouldn't say dominoes, just the steps for a cell to go through programmed cell death in nematodes. And then it's the same in every plant and animal on our planet, including in our bodies. There's a lot of this programmed cell death in our normal development. When we are fetuses, we have about 30% more brain cells than when we're babies. And they just get wiped out by programmed cell death because they didn't make the right connections. So it's kind of like a sculpting process, where you remove extra stuff. Between our fingers and toes, as fetuses, we had webbing initially, very early, and that gets wiped out to help create the individual identities. Name somebody who doesn't, ducks. Right? So there was some evolutionary change in ducks where they didn't wipe it out, and they ended up with webbing like that, Yeah?
It was kind of like off topic, but like not. So, like when your teeth are coming in and it's like pushing through like the gums,
Yeah.
Do you think there's programmed cell death in there in the gum tissue?
I don't know. Yeah, which I just have to say quickly as an aside, and maybe we'll have a early concept chat today, because it's nice to have it and then kind of keep going, have a break. And just as an aside, before I worked here, I worked in Seattle, I worked in a tall building, and so we would ride the elevator during the day. You have to go, when you're a scientist, you have to go get this, go get that, go get some ice, go do the centrifuge, and they're scattered throughout the building, and this was a tall, narrow building. So we'd go up and down the elevator. So I'd ride the elevator every day, kind of carrying my experiments with me, and I was a postdoctoral fellow, and there was a grad student named Mary. And so I got on the elevator with Mary, and I grew up in Minnesota, and in Minnesota, you say hi to everybody you meet. People in Minnesota drive their cars like this, and they wave to everybody as they drive by. So I got on the end of the elevator, and there's Mary. Hi, Mary. And Mary only said something to me twice, even though we worked in the same building for 5 years, like that. And she was frowning, like that. And so I, in my mind, nicknamed her Mary quite contrary, because she was mad about something like that. And here's one thing she said to me. She said to me, all of a sudden, I didn't even recognize, oh, you have a voice, like that. And we're in the elevator, I'm just like, and she says, you have male answer syndrome. And I'm like, what's that? And I don't know, but people know about that now. Like, if you need to ask for directions, you shouldn't ask a guy, because they're going to give you directions, even if they don't have a clue. They're just like, oh yeah, you just go by McDonald's, and you got to turn left, like that. Like that. They're just going to answer, even if they have a clue, right? So I was accused of having that, and consequently, it's like a relief for me to say, I don't know. I'd love to say that, because I'm like, Mary would be proud of me, you know, because I'm like, I have no clue, right? Do the gums above the little tooth buds, do they have to go through program cell? Yeah, I don't know. You know, and should I speculate? No. You know, I'm like, oh yeah, you go by McDonald's and sure, yeah, like that. So, yeah, I don't know. And, you know, is it embarrassing for me to do that? No, I love to say I don't know, right? It's one of my favorite things to do during the day is to be like, I don't know. Yeah, I know a lot of stuff about that. Just to say, the other thing Mary said to me once is she was on the elevator and she was holding like a worn out novel. And it was clear from the cover, I could see the way she was holding it. It was some kind of science fiction fantasy novel. And so I said to her, this is so great, and we gotta learn. I said to her, Mary, what's that book you're reading? And she looked at me scowling. Oh my God, it's so good. And she says to me, this is a book about the future where they clone out of humans the Y chromosome and get rid of it.
Oh.
So that's good. All right, let's take a look at this. We have a video. There's an nematode crawling. They're raised in lab on petri dishes like you use in microbiology. They eat bacteria. So scientists spread the bacteria on an agar plate like that, and these worms crawl across them, eating them, okay? And you really can't quite see them with your eye. The adult is almost visible to the naked eye, and so they're looking at them under a microscope, and they move this way. And they're just a really good model organism because they develop, it has about 1,000 cells like that, and if we pause it, it has another interesting feature in its look under a microscope, and that is, this is a light microscope. The animal is not stained, it's clearly alive, but you can see all of its cells. Those are cells right there. And you can see them all in the living animal, and you just look at them like that. And you can see them throughout the life. So you can watch that even without staining it with any tag. You could stain it and have a tag, and that's helpful. You just watch it. And it's visible because it's looked under a microscope that has a little kind of prism, so you can see that kind of shadowing. And this is what that microscopy is called. It's a regular light microscope, just with a little filter tube. Like a little prism, and we could just call it DIC. So the question on the exam could be 2-point questions. Why, give me 4 reasons why nematodes are excellent model organisms. 1, you can just look at the living animal under DIC and see all the cells. You know, most people don't call it DIC, most people call it Nomarski optic, but you don't need to learn that. I just put that up there so you know you're aware of that. Again, it's one of these things where there's too much of this in biology, but it'd be great. DIC, so that's one reason that nematodes are a model organism. And that's the first part of our story today, to get this set up to talk about programmed cell death. What's another reason that they are model organisms? Another reason they're model organisms is they're really good for genetic studies because the sexes are males and hermaphrodite. And a hermaphrodite is producing both eggs and sperm. So for geneticists, that's great because you can do a cross where you just cross the animal with itself. And I'll give you an example of that by going to that video and showing you. So in this animal, these bigger cells here are developing egg cells. The sperm cells are too small, but this is a hermaphrodite because there's no females. This is a hermaphrodite, and there's sperm in here, and right there is the opening where the eggs would be released. And usually the animal releases the eggs before they're fertilized by itself, right? Because it's trying to have sexual reproduction. And this little opening is talked about in the book, and that's the basis for that figure we don't need to learn about. That little opening is called the vulva, and developmental biologists made a lot of great discoveries of these genes by studying mutants where they would not have a vulva, and so they couldn't lay the eggs, and so that wouldn't kill them because they could have fertilization inside their body because they had sperm and eggs. And so that's a tricky thing in development is a lot of times you make a mutation in a really interesting gene and it's gonna, they're gonna be dead. And you really can't study something, you can't propagate it, it's just dead, right? It's lethal. And so these mutations where the vulva was not formed were a problem with the pathway of development, no vulva, and the animal did not die. It reproduced, and you could propagate it. And the mutant phenotype is famous. All these people are like me, and if you knew Ken Weber, there's weirdos. The mutant phenotype is called bag-o-worms because they can't get out, they hatch, and the scientist just pokes the dead animal, and a bunch of babies come out like that. So just a little side note about that. So model organism, you know, because you can see it and study it like that really easy with DIC. Then it is a hermaphrodite, so the genetics are really different than like a fruit fly or a zebrafish or a human like that. You can do a lot of genetics really fast like that. And then I mentioned they're easy to grow, you know, in a dish, in a lab. You can freeze them, thaw them, they're alive. So it's just a model organism for lab research, okay? So here we are, and oh, we're gonna do that, sorry, forgot. Let's do a concept chat. And so that's where try to find somebody else. So you can stand up and walk and go talk to Jesse hiding in the corner or anybody else. And we're gonna try to have just 2 people. As soon as it's 3 people, it's 3 people to talk, and we do it fast. So just find 2 people. The only reason we'd have 3 is that there's an odd number of people in the class. Okay? So I'll try to help you if I see 3 people and 3 people and say, look, you guys go. So just team up, and you're gonna talk about 3 things that you thought were important today. In just a couple of minutes. All right, here we go. So we'll do this another minute. Yeah, and because if you're like, oh, we're all set, you know, you can talk a little bit about like what your other courses are and stuff like that. Because part of this is, you know, we scramble to get it done, but we're also getting a chance to, you know, meet classmates like that a little bit. That's great. It's also great because then I get to start learning people's names, like Brianna. I don't know Brianna. Hi, Brianna. So what is one thing that your group talked about that you thought was important? Oh, now we have to listen to Brianna. Woo! Woo! Brianna. And let me just mention that tomorrow I have to go get my hearing checked, because I attended so many concerts, I can't hear. So like when Brianna starts talking and everybody else is talking, I'm just like, yay, you know. So yeah, it's a little bit embarrassing, but I can't hear very well, whatever, you know. So Brianna, what's one thing that you 2 talked about?
We talked about how stem cells have asymmetric division, where they divide into one stem cell and then one precursor cell, which can then differentiate into something more specific.
Awesome. Yep. And so that's a really key thing. Unequal cell division in stem cell biology, they refer to it as asymmetric division. The general term, the old school term, is mosaic development. It's only one of 2 ways that cells can become different on our planet. And then let's see, I also circled Nick.
All right. One thing that we talked about was terminal differentiation, which is the end of the road for differentiating cells, such as like muscle cells will stay as muscle cells, but to reverse that, you can take stem cells
Yeah, yep, terminal differentiation, yep. And that is true, like that. It's definitely a big part, big centerpiece of biology, right? We need those cell-like stem cells because the muscle has undergone terminal differentiation, right? That's like classic dogma. Stem cell biologists are just reversing it, literally, right? Through experimental manipulation. And we talk about it because it's significant, but it also helps to illustrate the biology. A great proof of genomic equivalence is an induced pluripotent stem cell, right? Oh, I could take any of your cells right now and turn it into a stem cell and build a new allele, right? Just, right? And of course, stem cell biologists haven't got that far. Quick example of that, down at MHIR, Maine Health Institute for Research, where the scientists are just doing this kind of stuff, and we have a dozen interns down there right now, like Chloe and Mohammed and others. They're doing that kind of research. And he doesn't work there anymore, but there was a stem cell biologist about 10 years ago that was working down there, Joe Verdi. And in the developmental biology lab course, we go down there on a field trip and we will meet briefly with some of the scientists. So here's Joe Verdi, stem cell biologist. And stem cell biology is like in the Wild West where there are no laws, you know, because they're just like, we could try this, we should try that, like that. And of course there are laws and rules, but it's more kind of like a cowboy Western style. So he's a cowboy like that. So we go in to talk with him, and I knew what he would say, but I said to him, in our lecture course, Joe, we've spent the semester learning about concepts like terminal differentiation, and you'll see we'll learn other terms like determination, commitment, specification. So I rattle off half a dozen terms that are classic truth in embryology, developmental biology, right? And I said, you know, from a stem cell biologist perspective, what do you think about that? And these are terms, you know, that are going to be worth a bunch of points on our exams. And this cowboy stem cell biologist, Joe, says, I think they're a bunch of horse shit. You know? Like that. Because of that, you know, what he's doing and they can do, right? So all of what he can do is it's experimental manipulation, right? Yamanaka, right? Why aren't we building you a new kidney yet? One of the transcription factors helps get the cell cycle activated to make an induced pluripotent stem cell. That transcription factor is a very famous oncogene. So yeah, we can make induced pluripotent stem cells, but when we say, yeah, we're gonna get these cells growing again, could that become cancer? So, you know, it's like the cowboy thing where you're like, yahoo! You know, we're gonna shoot up this town. And we're like, Joe, take it easy, cause that's gonna cause trouble, you know, and stuff like that. So it's like that. It's really fascinating that we'll learn core truths of that nature and how things are developing. And then there's this modern piece where you're like, I'm glad to know that core truth so I can appreciate the challenges and successes of this modern regenerative medicine. Okay, so I wanted to show you this slide because lots of you would have learned this term apoptosis. And that's a kind of programmed cell death, and it's taught in a bunch of classes in college. And it turns out that there are really two major types of programmed cell death. One is apoptosis, one is autophagy. Autophagy is really common in development. And they're quite different. Same outcome, a cell's gonna die, but they're really quite different. So if you've heard about apoptosis, yeah, we're talking about that with programmed cell death. But developmental biologists really tend to say programmed cell death or PCD when they're talking about it. So we'll say PCD. The example that the nematode people were working on, that was really an example of apoptosis. Great. Yep, just put that up there. Okay, so now let's just show you this. So here's a picture of the three scientists that were given the Nobel Prize in 2002 for this work on programmed cell death. And I will bring up names and times and places, but that are not important. You don't need to learn them, right? So, you know, Seymour Benzer, John Tolston, and Horvitz, Bob Horvitz, like that. You don't need to worry about it. The reason I show you this picture is because it can help you remember stuff, right? So these three share this Nobel Prize, and they're doing what they're famous for in this picture. So Seymour is the guy in the 1960s who's like, we need another model organism, maybe a hermaphrodite. So he's the leader. Then there's John Sulston, look at him, right? So he looked at under a microscope with these DIC optics, the shadowing kind of optics, at nematodes, and watched them grow and develop. And he watched every cell, and he mapped it, and he discovered that the cell lineage, that pattern of cell division in nematodes, let's me find the term. They go through an invariant cell lineage, and that just means that each animal develops the same way, the same pattern of cell divisions. We don't do that, other animals do that. It's just another odd feature that made this a fantastic model organism. And that is, you can't see this too well, but we're gonna zoom in on it. What's shown here is on the left is the fertilized egg, and then it divides again, and he just mapped out all the cell divisions. You see there's somewhat of a timeline from left to right. So this is like a fertilized egg, and then an early baby nematode, growing, adult nematode. And so that guy, again, name not important, John Sulston, he looked and mapped those all out. And what you see when you zoom in on it is, let me just show you what we're zooming in on. So this is all of them. We're gonna produce an adult with about 1,000 cells, and they're always the same pattern. And so we're just gonna zoom in on this little area on the next slide. And it's some of the cell divisions, and it's early cell divisions in the development. So it's a little baby nematode, and look at what he saw. Here are these cell divisions he's mapping them, and these X's are the cell just dies. That cell at that time just dies by programmed cell death. And its job is done. Sometimes cells play a kind of guiding cell for another cell, like a nerve cell that's growing, and the cell's just gonna guide it, and it's done. And so at that time of development, it dies. So that's what he saw. And see, there's a bunch of it, and they're very specific. And what could happen then is that, I got our time down here, that clock is running slow, is that he's the starting point to do experiments. Now, let's do some mutations and find some mutations where one or more of those cells don't die. And then they must have a mutation in a gene that's necessary for programmed cell death. So what is the gene? And the third guy in the picture, that's his role.
So what are the ends that don't have X's? Are those
Those still live. They live for the life of the embryo.
Okay.
And they might be stopped. They're not going to divide anymore, but they're just alive.
Gotcha.
Right, like that. So see the third guy leaning forward? He's a professor at MIT, and his lab did the genetics. And they found all the mutants, and that's what we really talk about, is a little bit of genetics of, well, what does it mean to find mutants and define which genes are important and needed for cell death, and what is their role, like that. And his lab did it all. He has like 40 students in his lab, undergrads, grad students, postdocs, technicians, all at MIT, and they work all the time except when there's a Red Sox home game, literally. And we can kind of relate to them. They're just a bunch of Red Sox fans. And MIT is not far from Fenway, so they put stuff on ice and they go over to Fenway, and it's a lot of people. And so he's the one who did the work, right? A leader, let's get this going. A guy who did the background stuff and set this up. That was their contributions. And then Bob's lab just does this. And so next Tuesday, we're going to be ready, and we'll talk about the quiz, any questions you have, but we'll be talking about this and the genetics of it, because we want to get a little genetics into this, because that's a big part of modern developmental biology. Great.